Good morning, dear students. Last class, I have discussed about principles of inheritance and variation. Principles of inheritance and variation. So, in this chapter, I have discussed some important points. What is genetics? And what is heredity? What is inheritance? And what is variation? Who coined the term genetics? So, in this topic, I will discuss what is genetics and what is inheritance and variation and what is heredity. And the Greco-German selected the pea plants for his experiments. Why? He selected pea plants only for his experiments. And Mendel selected 7 pair of contrasting characters. 7 pair of contrasting characters. All those things I will discuss in the last class. So, the branch of science which deals with inheritance and variation. The branch of science which deals with inheritance and variation is called genetics. This branch is called genetics. The term genetics was coined by Bateson. Bateson very first coined the term So variation means the characters inheritance. Inheritance means the parental characters are transferred to their offspring. Transfer of parental characters to their offspring is called inheritance. And variation is differences. The offsprings are not alike to their parents. The offsprings are not exactly alike to their parents, but they show differences. The offspring shows the differences called variation. Variation is differences, not exactly alike. The offsprings are not exactly alike to their parents, but the offspring shows the variation is changes or differences. Variation is difference. The offspring shows the differences is called variation. So what is heredity? Heredity means the offsprings resemble their parents. The offsprings resemble their parents is called heredity. So the very first in the field of genetics, so Gregor and Mendel worked very first in the field of genetics. There, Gregor and Mendel worked very first in the field of genetics. Next, genetics. So Gregor and Mendel was born in Moravia in 1882. He born in 1882. And Gregor Mendel worked on Pisum satarum. He working on Pisum satarum, garden pea plant. He worked on Pisum satarum, garden pea plants about 7 years. About 7 years. And his works and his results were presented. His results are presented in 8th February 1865 to Society for the Scientific Society for the Scientific Study in Rome. This means the results are published in 8 February 1861, Society for the Study of Scientific, Society for the Scientific Study in Blue. And unfortunately, the work of Mendel was unnoticed. Mendel work was unnoticed by the scientific world, and Mendel died in 1884. And after the death of Mendel, he was awarded as the father of genetics. And the three scientists, Euclides, Karl Lorenz, and Teschmark, these three scientists can rediscover all the principles of genetics proposed by Mendel. All the Mendel work once again rediscovered by Euclides, Karl Lorenz, and Teschmark. Okay? And then Mendel selected the pea plants for his experiment because the pea plants are easily cultivated, not required aftercare. Pea plants are annual, they give results within a year, and pea plants are bisexual. Pea plants are bisexual. The self-pollination is conducted or carried out, but cross-pollination in pea plants made by manually. In pea plants, cross-pollination is conducted by manually. And the pea plants, true are pure line breeding variety with observable pair of contrasting characters. So pure line breeding variety with observable pair of contrasting characters. By all these reasons, and the hybrid plants are fertile and they produce the seeds. The hybrid pea plants are fertile and they produce the seeds. By these reasons, the Mendel has selected the pea plants for his experiments. So, among the various contrasting characters are observed in the pea plants. Various of different types of contrasting characters are observed. But among these, Mendel selected only 7 pair of contrasting characters. Mendel selected only 7 pair of contrasting characters for his experiment, like stem height, flower position, flower color, shape of the pod, position of the pod, and then seed shape and seed color. These are the 7 contrasting characters selected by Mendel for his experiments. Up here I discussed last class. So terminology, I have discussed terminology. The terms, some important terms used in the genetics. First one, true or pure line breeding variety. Pure line breeding variety. So the variety, what is true or pure line breeding variety? The variety in which the offspring is produced. The variety in which the offspring produced. The variety in which the offspring produced. Having parental tracts. Having parental tracts. Having the parental tracts. With variety is one in which the offspring is produced. The variety in which the offspring produced have parental tracts on continuous self-pollination. On continuous self-pollination. or continuous self-pollination. Continuous self-pollination is called pure line breeding variety. Pure line breeding variety. For example, tom tea plants. Tom tea plants. On continuous self-pollination, they produced tom plants only. In all generations. In all generations, they produced only tom plants because tom tea plants are continuously self-pollinated. Here, this is about pure line breeding variety. The variety in which offspring is produced, they have parental tracts. Traits means character. Their possessors, they have parental tracts. On continuous self-pollination is called pure line breeding variety. For example, tom tea plants are continuously self-pollinated. They produce only tom tea plants. Continuous self-pollination. That's why they produce tom tea plants only. It's called pure line breeding variety. Second one is contrasting character. Second terminology is the contrasting. Second one, contrasting character. Contrasting character means alternative expression of the same character. Alternative expression of the same character. Is called alternative expression of the same character is called contrasting character. Alternative expression of the same character is called contrasting character. For example, the flower color occurs in two contrasting forms like violet and yellow. Violet and yellow. Yeah. So alternative expression of the same character is called contrasting character. The flower color, for example, the flower color has two contrasting. They appear in two contrasting. So violet and yellow, two contrasting colors, violet and yellow. So next one is alleles. Next term is alleles. So alleles is defined as alternative form of gene. Allele is an alternative form of gene. for alternative expression of alternative expression of same character, alternative expression of the same character present on same loci, present on same loci, present on same loci on homologous chromosomes, on homologous chromosomes, homologous chromosomes, homologous chromosomes. Here, see alleles. Alleles is nothing but alternative expression, alternative form of gene, alternative form of gene for alternative expression of the same character are present on same loci, on locus, on homologous chromosomes is called alleles. For example, a gene controlling the height of stem, the gene controlling the height of stem or the genes which are controlling the height of stem. For example, T for tallness and small t, it controls dwarfness. Both are the capital T, which controls the tallness, and small t controls the dwarfness. These are present on the same loci, on the homologous chromosomes. Height of the cell can be controlled by two different forms of genes. For example, capital T so controls tallness and small t controls dwarfness. Both are present on the loci, on homologous chromosomes. But alleles are nothing but alternative form of gene, alternative form of gene to control alternative expression of the same character present on same loci, on homologous chromosomes. Next one, fourth one. Allelomorphs. Next, allelomorphs. Allelomorphs. Allelomorph means a pair of contrasting character of an organism. A pair of contrasting character. Pair of contrasting character. Contrasting character of an organism is called as allelomorphs. A pair of contrasting character of an organism is called as allelomorphs. For example, shape of the seed, in shape of the seed, inflated and constricted, or color of the seed, yellow and green. So these are pair of contrasting character. Pair of contrasting character of an organism is called as allelomorphs. So best example, for example, color of the seed, yellow and green. These are the contrasting characters of an organism. Next, homologous chromosomes. Homologous chromosomes. Homologous chromosomes. Homologous chromosomes. Homologous chromosomes. So what are homologous chromosomes? which are alike in all aspects. Which are alike in all aspects. A pair of homologous chromosomes. Pair of homologous chromosomes. A pair of homologous chromosomes which are alike in all aspects. Which are alike in all aspects. Are called homologous chromosomes. Are called homologous chromosomes. A pair of homologous chromosomes which are alike means similar or same in all aspects. Among these, one pair is derived from. Among these, one pair is derived from mother. One pair is derived from mother. It is called maternal chromosome. Called maternal chromosome. Next, another pair. Derived from father. It is called paternal. Is called paternal. See, homologous chromosomes are alike in all aspects. Among this, one pair is derived from mother, called maternal. Another pair is derived from father. It is called paternal. This is called homologous chromosomes. Next, hybrid. Hybrid. What is hybrid? What is hybrid? Hybrid is an individual. Hybrid is an individual obtained from cross between two parents. Obtained from cross between two parents of same species, but differing in one pair of contrasting character, or differing in characters, differing in contrasting character. It is called hybrid. Clear? What is hybrid? Hybrid is an individual. Hybrid is an individual. Hybrid is an individual. It is obtained from crossing between two parents of same species, differing in contrasting character. Is called hybrid. Here, hybrid is an individual. It is obtained by crossing between two parents of same species. These two parents are belongs to same species, not different species, same species. Differing in contrasting character is called as an hybrid. Next one. Next one is phenotype. Next one is phenotype. Next one is phenotype. What is phenotype? The external appearance of an organism. External appearance of an organism is for a particular character is called phenotype. Phenotype is nothing but external appearance. External appearance of an organism for a particular character is called phenotype. Phenotype is defined as external appearance. External character. Appearance means character. External appearance of an organism. Of an organism for particular character. For particular character. Particular character is called as phenotype. Here, next, genotype. What is the genotype? Genotype. See, genotype means, what is genotype? So, genotype means an allelic pair of a gene in an allelic pair of a gene. Allelic pair of a gene controls allelic pair of a gene controls particular character of an organism. Particular character of an organism is called genotype. Genotype is also defined as genetic constitution of an organism. Genetic makeup or genetic constitution of an organism or genetic makeup of an organism is called genotype. See, genotype is defined as in an allelic pair. In an allelic pair of a gene. Allelic pair of a gene. In an allelic pair of a gene controls particular character of an organism. Is called genotype. Clear? It is also defined as genotype is also defined as genetic. It is also defined as genetic constitution. Genetic constitution of organism. Genetic constitution of an organism is also called as genotype. Here, phenotype only external appearance. Genotype is internal. That means a gene can control a particular character of an organism. It's called genotype. Phenotype and genotype. Next. Phenotypic ratio. Phenotypic ratio. Phenotypic ratio. Phenotypic ratio. Phenotypic ratio. The ratio between phenotypic ratio is defined as phenotypic ratio. Phenotypic ratio is defined as the ratio between individuals of F2 generation. Individuals of F2 generation is called phenotypic ratio. Phenotypic ratio. The ratio between the individuals of the ratio between the individuals of F2 generation. F2 generation is called phenotypic ratio. Phenotypic ratio. Ratio between individuals of F2 generation. Phenotypic ratio. Like what is genotypic ratio? Genotypic ratio. Genotypic ratio. So genotypic ratio is the ratio between genotypic ratio, the ratio between the individuals of F2 generation. F2 generation is called genotypic ratio. So genotypic ratio is the ratio between individuals of F2 generation. The ratio between the individuals of F2 generation is called genotypic ratio. See, the phenotypic and genotypic ratio. The next one, next terminology is homozygous means similar. Homo means similar. In an allelic pair of gene. Allelic pair of gene. The allelic pair of genes are similar, or nothing but similar allelic pair of gene. Similar allelic pair of gene. Is called homozygous. Similar allelic pair of gene is called homozygous. So the homozygous are called homozygotes. They are called homozygotes. Because they produces only one type of gametes. They produces only one type of gametes. For example, capital T and small t. So similar, they produces only one type of gametes. Next, heterozygous. So heterozygous means dissimilar allelic pair of a gene. For pair of contrasting character. Heterozygous, dissimilar allelic pair of gene. Dissimilar allelic pair of gene for a pair of contrasting character is called heterozygous. These are called heterozygotes. They produces heterozygotes. They are called heterozygotes because they produces dissimilar gametes. For example, capital T and small. Hetero means different or dissimilar allelic pair of a gene called heterozygous. The individuals are called heterozygotes because they produce dissimilar gametes. For example, capital T and small. Dissimilar allelic pair, they produce different or dissimilar gametes. Yeah? Next one is dominant G. Dominant G. The G in a allelic pair, the G can expresses in an allelic pair. Allelic pair. The G can express. Is called dominant G. Dominant G means it expresses. Dominant G means it expresses in allelic pair. This is called dominant. And recessive G. Recessive G. Recessive G. In an allelic pair, the gene fails to express. Is called recessive G. In an allelic pair, the gene fails to express into pair of contrasting character is called recessive G. Means dominant G means it expresses. Recessive means the G fails to expresses its character out of two contrasting character. This is called dominant G and recessive G. Next one is dominant character. Dominant character. See, dominant character is the character which expressed. The dominant character is one. It is expressed or appeared. The character appeared. Is appeared out of two contrasting character. Out of two contrasting character. Is called dominant character. The character which is appeared. Out of two contrasting character is called dominant character. Next one, recessive character. So the character fails to appear. Out of two contrasting character. Recessive character is one which fails to appear out of two contrasting character. It's called recessive character. Recessive means does not express. Recessive means suppressing. It does not express in the presence of dominating. That's why recessive character fails to express or appear out of two contrasting character. Recessive means suppressed. So this is called dominant character and recessive character. So last one, checkerboard or Punnett board. Last one, checkerboard. So checkerboard is the schematic table. It is the schematic table to Checkerboard is the schematic table which is used to determine various combinations of phenotypes and genotypes of offsprings. See, checkerboard is the schematic table which is used to determine various combinations of phenotype and genotypes of offsprings. Very first, checkerboard was discovered by R.C. Punnett. This board was discovered by R.C. Punnett. That's why it is also called Punnett square. That's why it is also called Punnett square. Discovered by R.C. Punnett. That's why it is also called Punnett square. See, these are the different terms commonly used in the genetics. These are the different terms commonly used in the topic genetics. So it's finished. These are the common terminology or common terms used in the genetics. Next class I will discuss about monohybrid and dihybrid cross. Okay, thank you.